So in this part of the lecture, we'll cover glomerular filtration in a little bit more detail, and we'll focus on the glomerular filtration rate and how glomerular filtration is regulated. So from the earlier lecture, you should know by now what the function of the kidney is. And the kidney basically filters. Filtration basically involves transfer of a lot of components like water and waste into the glomerulus from the blood. Okay, and once everything is filtered out from the blood into the glomerular capsule, as it passes through different parts of the nephron, a lot of important substances are reabsorbed back into the bloodstream because the nephron is surrounded by peritubular capillaries. So all of this nephron, all the parts are surrounded by a whole bunch of capillaries, and they constantly function in reabsorption of anything that is required by the body. So what you're looking at right over here is the Bowman's capsule that you are aware of, and this portion of the Bowman's capsule is called the juxtaglomerular apparatus. Now the juxtaglomerular complex or juxtaglomerular apparatus is a portion where you find the afferent arteriole that brings blood to the glomerulus, and then you see the efferent arteriole that takes away blood away from the glomerulus after a lot of substances have been pushed through into the glomerular space. And this over here is actually a part of the loop of Henle. So you see, this is what a nephron would look like. So that is a nephron, this is another nephron, and you can see as the loop of Henle descends and then ascends again, a part of it articulates very closely with the glomerulus, and that is the part that you're looking at right over here. Okay, so all these portions around, all these tubules around the glomerulus comprise of the glomerular apparatus. Now there are three main components of the glomerular apparatus that regulate filtrate formation and the blood pressure as well, blood pressure of the body and the blood pressure of the glomerulus as well. So what does filtrate mean? So the process of filtration occurs in the glomerulus, wherein a lot of fluids are filtered from the blood. And remember, there are a lot of large perforations. You have perforated capillaries over here that have fenestrations. So a lot of substances can be pushed through the podocytes and the fenestrations over here into the glomerular space, and a lot of its contents later get reabsorbed into the peritubular capillaries later. So filtration occurs at the membrane in the boundary between the glomerulus and the Bowman's capsule, and this is largely controlled by a whole lot of cells in the juxtaglomerular apparatus. So the macula densa is a part of the juxtamedullary apparatus, but it is really located in the ascending limb of the loop of Henle. So when this loop comes back up, it articulates closely with the Bowman's capsule, and this is where we have a really thick, dense layer of cells, specialized cells known as macula densa. The main role of macula densa is to act as chemoreceptors. Chemo means chemical. So the main job of macula densa is to monitor the filtrate's sodium chloride content or salt content as they enter the nephron. Okay, because water follows sodium. Okay, so the concentration of sodium in the body is very important in determining how much water we retain in the body. Okay, and then the other component are the granular cells. So the granular cells are present right over here in the the efferent arteriole. So these cells contain large granules that secrete renin enzymes. So if you remember the renin-angiotensin aldosterone mechanism, which is very, very important in regulating the blood pressure. We looked at it in the respiratory system, and before that we looked at it in the cardiovascular system as well. Okay, so this is where renin enzyme is secreted. Renin enzyme basically kickstarts the whole renin-angiotensin aldosterone mechanism. Okay, and the cells that secrete renin are known as the granular cells. And the other important part of the juxtamedular apparatus is the extraglomerular mesangial cells. Now, the extraglomerular cells are basically the cells that connect the macula densa to the granular cells, then help them communicate. So right over here, you have a whole bunch of gap junctions that pass signals in between the macula densa and the granular cells. So let's quickly preview glomerular, the different stages of urine formation. First one is glomerular filtration, where the fluids, the blood basically is pushed through, but the blood cells are too big to pass through. So all you have entering the Bowman's capsule is a whole lot of water, not very much proteins, no glucose, some solutes. Okay, so glomerular filtration basically creates plasma-like filtrate from the blood. Okay, so this portion will not have any blood cells in it, should not have any blood cells in it. Okay, and the next stage is tubular reabsorption and tubular secretion. Tubular reabsorption is basically the stage wherein useful solutes are removed from the filtrate and taken back into the peritubular capillaries surrounding the nephron. Okay, so at this stage the body reevaluate what it needs, and the body says, hey, I need more sodium or hydrogen, and some sodium or hydrogen may be taken up in this area, right? And tubular secretion is the process wherein any unwanted substance from the capillaries is put back into this filtrate that will later form urine. So it's very important that you understand the difference between reabsorption and secretion. During reabsorption, anything that is necessary for the body is reabsorbed into the capillaries, whereas tubular secretion is the process where additional unwanted substances is secreted back into the filtrate that will form the urine. And the last stage is water conservation. So the body again reevaluates. There are three important hormones that we talked about in the previous lecture that it was posted. And water conservation is basically the last step, and it takes place in the collecting ducts. And one of the hormones that is very important in conserving water is antidiuretic hormone. Based on whether we need more water or not, a lot of water will be absorbed by the body or secreted or just left into the filtrate. So this is again another picture showing formation of urine. This is the afferent arteriole. These things have moved around, but this is the afferent arteriole. This is the glomerular capsule. Peritubular capillaries are the capillaries surrounding the nephron, and this red arrow signifies tubular reabsorption. And the blue arrow signifies tubular secretion into the tubule. So this is the tubule of the nephron, and things are either taken up by the capillary with the help of a process known as tubular reabsorption, or things, unwanted waste, are secreted back into the tubule with the help of tubular secretion. So glomerular filtration is governed by or driven by hydrostatic pressure, meaning the pressure exerted by the fluids. And the other thing that affects glomerular filtration is osmotic pressure. Okay. Now the glomerulus is a very efficient filter because the membrane is really very permeable because it has a very large surface area, and the pressure in here is significantly higher than other capillaries. We'll see in a minute why. Now the filtration is due to the fenestrations in the glomerulus, and the molecules that are larger than 5 nanometer are not filtered. So large proteins, blood cells, these cannot pass through the fenestrations. So the concentration of proteins function in maintaining the colloidal osmotic pressure of the blood that determines the glomerular filtration rate eventually. Okay, so glomerular filtration rate is of a lot of clinical interest because glomerular filtration rate tells us how well our kidneys are functioning. So the glomerulus produces a filtrate that is free of cells, that is free of proteins. It's not a good thing to see when I say filtrate. It eventually becomes urine. So you really do not want to see any red blood cell or any proteins in the urine. So most fluid that is filtered through the glomerulus and enters the glomerular capsule eventually is reabsorbed back into the bloodstreams. Okay, so 80% of the liquid is reabsorbed back into the bloodstream. The outward pressure that affects filtration due to the fluids in the capillaries right over here is called hydrostatic pressure. So the fluid pressure is called hydrostatic pressure, and the fluids are basically contained in a tuft of capillaries in the glomerulus. So the hydrostatic pressure is basically the same as the blood pressure in the glomerulus. Okay, so the BP in the glomerulus is about 55 mm of mercury, and in other capillary beds, I think I should write that down. Here, so the other capillary beds only have a blood pressure of 26 mm of mercury, and why do you think this is the case? If you look at the afferent arteriole, and this one over here is the efferent arteriole, you can see the difference in diameters of the afferent and efferent arteriole. And because you're bringing in a lot more blood through the afferent arteriole, but the blood that leaves the glomerulus is forced through a very small arteriole that is tinier in diameter than the afferent arteriole, right? So you're pressuring, applying a lot of pressure in forcing a lot of blood out of the efferent arteriole that is smaller in diameter, right? And that's why the pressure inside the glomerulus is higher than that of the surrounding arteries. Okay, now after filtration has completed, the capsular space collects the filtrate, and this filtrate practically does not have any proteins at all, because of which the osmotic pressure in the capsular space over here is nearly zero. Now osmosis is basically, or osmolarity is basically determined by the amount of solutes in a solution, and in this case the filtrate does not have any proteins. Okay, and because of that, the osmotic pressure is very, very close to zero. So in this slide, just make sure you focus on net filtration pressure and how you get the net filtration pressure. So there are a lot of forces acting in the Bowman's capsule. You have the blood pressure, then over here a lot of colloids are, stuck at the pores, they cannot pass through. So you have a lot of colloidal pressure exerted by the molecules that are stuck in the concentrated blood over here before they can be passed through. And then you have the fluid pressure of the blood. And then you have some pressure in the capsular space, right? So all of these act and contribute to the net filtration pressure in the glomerulus. Okay, so let's look at it. So hydrostatic pressure basically means fluid pressure. Okay, and due to the pressure, red white blood cells, platelet, albumin, large proteins, they're too large to pass through these pores and they remain in the capillary. So in this area where you have a lot of blood cells and large protein molecules trapped, causes the average osmotic pressure within the capillary to be slightly higher. So it's about a 32 mm of mercury, to 30 to 32. So COP stands for colloidal osmotic pressure. And in the absence of protein in the Bowman's space or the capsular space results in nearly zero osmotic pressure, because this area does not have any protein in it. Okay, and then you have the capsular pressure, or CP, over here. So the capsular pressure is basically the capsular hydrostatic pressure. The capsular hydrostatic pressure, or the capsular pressure, one and the same thing, is the pressure exerted against the filtration membrane by this filtrate that is in the Bowman's capsule. Now we're talking about hydrostatic pressure over here and not osmotic pressure, because the osmotic pressure in here is zero. But there are fluids in here. So these fluids exert a capsular hydrostatic pressure of about 18 mm of mercury in here. Okay, so you have the blood hydrostatic pressure trying to push out to force solutes and liquids into the capsular space, and then you have the colloidal osmotic pressure trying to move away because they cannot pass through the filtrate, the pores of the filter. And then you have the capsular pressure that is exerted by the liquid in the capsular space, again trying to exert some pressure against the glomerular capillaries. So the net filtration pressure finally amounts to approximately 10 mm of mercury that is exerted on this area. Okay, and let's see, the hydrostatic pressure in here, the net hydrostatic pressure is sufficient to push water through the membrane despite the osmotic pressure working against all the pressures in here. And the sum of all the influences, both osmotic and hydrostatic pressure, results in the net filtration pressure. Okay, and finally the net filtration pressure is about 10 mm of mercury, and this pressure differs in different areas of the capillary. So in the arterial end, obviously it can withstand significantly more pressure. So in the arterial end, the net filtration pressure is about 10 mm of mercury, and in the mid capillary region, basically the area where there's no net movement of fluid, since the capillary hydrostatic pressure is equal to the colloid osmotic pressure right over here, the net filtration pressure in the mid capillary region is zero, so there's no net movement of fluid. And then you have the venous end. Okay, now arteries follow veins, so wherever you find arteries, you will find veins. So these capillaries also have a venous end, and at the venous end, the net filtration pressure is negative 7 mm of mercury, because in the venous ends of the arteries is where you find a lot of reabsorption taking place into the blood. Okay, so in the venous end, the hydrostatic pressure is less than the blood colloidal osmotic pressure. Okay, and that amounts to a negative net filtration pressure. Okay, so for your purposes, understand that the net filtration pressure is 10 mm of mercury, and understand why that happens, and understand what are the forces that causes the net filtration pressure to be 10. Clinically, what is of interest to us is glomerular filtration rate. The glomerular filtration rate is the filtrate formed per minute, and the glomerular filtration rate can be determined by using the net filtration pressure and the filtration coefficient, which is Kx. So this is a constant, and NFP is the net filtration pressure. Now, on an average, the normal GFR for a male is 180 liters per day or 125 ml per minute. For a female, it is about 105 ml per minute or 150 liters per day. Now, depending on the size of a person, for a petite person, the GFR may probably be lower. So there's a different number for each person. However, just keep in mind, when things are filtered through the glomerulus, 99% of the filtrate is reabsorbed, and in the end, you only get 1 to 2 liters of urine. Now, along with GFR, what they look at is creatinine. Creatinine is also a good indicator of kidney function because in the kidney, in the urine, we excrete some amount of creatinine. Creatinine is a byproduct of creatinine phosphate that is produced by muscle, and so we have some amount of creatinine in the blood. When the levels go up, they end up depositing in the body. So creatinine along with GFR is a good indicator of kidney function. Now, if your GFR stands for glomerular filtration rate. If your GFR is really low, it means your kidneys are not working as well as they should. So older people above 70 will have a lower than normal GFR because GFR decreases with age, and as they age, their kidney starts malfunctioning because the cells start degenerating, right? So older people will have a lower GFR level. Along with GFR, the amount of creatinine we secrete in the urine will tell whether our kidney is normal or not. Now, if the GFR is lower than 60 and it persists for about 3 months or more, it indicates a chronic kidney disease. Okay, so when GFR is below 60, it indicates chronic kidney disease if it lasts for more than 3 months. Now, when the GFR is below 15, let me write this down. Yeah, so if the GFR, where's my pointer right now? If the GFR is below 60 for over 3 months, it indicates chronic kidney disease. But if the GFR is below 15, it is an indication of kidney failure. Now, this does not tell us the cause. It just tells us what exactly is happening, okay? Now, if the GFR increases, it leads to increase in urine output, right? Because there's a lot more pressure on the glomerulus. So this can cause excessive loss of water and dehydration. Along with water, obviously, we lose a lot of ions, so it can lead to electrolyte depletion. And if the GFR is really, really low, base are retained in the body, okay? So we end up retaining urea and nitrogen. And azotemia is the condition wherein we retain excessive nitrogen in the blood because it's not efficiently removed through the urine. The GFR is controlled by adjusting the glomerular blood pressure by 3 different mechanisms: autoregulation, sympathetic control, and hormonal mechanism. Now, we have looked at hormonal mechanism in the past, and we'll look at some intrinsic and extrinsic control mechanism that affect GFR and control GFR. So the glomerular filtration rate is controlled by 2 different types of mechanisms: intrinsic and extrinsic. Intrinsic mechanism involves everything around the nephron, like the juxtaglomerular apparatus locally within the kidney that controls the glomerular filtration rate. Keep in mind that the glomerular filtration rate is really a reflection of the pressure of liquids in that region. So glomerular filtration is affected by the main arterial pressure as well. And extrinsic control takes over when a lot of stress is involved. So extrinsic control involves the regulation by the nervous system and the endocrine mechanisms, along with the hormonal mechanisms like the renin, angiotensin, and aldosterone mechanism. So the first mechanism, intrinsic, mostly involves a myogenic mechanism. Myogenic mechanism is based on how the arteries and arterioles constrict or dilate to increase and decrease the blood pressure within the blood vessel, and this involves the contraction and relaxation of smooth muscles. Okay, so when the blood pressure increases, the smooth muscles constrict the afferent arteriole and they restrict the blood flow to the glomerulus. Okay, and on the other hand, they dilate the efferent arteriole. So this protects the glomerulus from extreme damage, right? So when there's a lot of blood pressure, you want to prevent overstretching and damage to the glomerulus because it's a really high blood pressure. So when the blood pressure is low, the afferent arterioles get dilated and they help in maintaining normal glomerular filtration rate despite fluctuations. Okay, so when the blood flow increases, the smooth muscles contract in the afferent arteriole and dilate in the efferent arteriole. Now when the blood flow is low or when the blood flow decreases, the afferent arterioles dilate to maintain normal homeostasis despite fluctuation. Now, the stable blood pressure in a normal person is in between 80 to 170 mm of Hg, which is that is the systolic blood pressure, right? Systolic is what we are mainly interested in when you're looking at the blood pressure. Now, low blood pressure also affects a lot of different mechanisms. If you remember, if the blood pressure is too low, way low is when you need to worry about it, because if the blood pressure is too low, it affects the intracranial pressure and it can cause ischemia in the brain, and lack of oxygen for a prolonged period can cause irreversible damage. Alright, but for glomerular filtration rate, really we are concerned about the systolic pressure, which can cause hemorrhage. Now, GFR needs to be kept relatively constant in order to maintain homeostasis. However, sometimes if there's a hemorrhage, it can cause a seriously low blood pressure in the glomerulus, right? So when there's a hemorrhage, there can be excessive loss of blood and it can cause a hypovolemic shock. So a hypovolemic shock occurs when you lose a lot of extracellular fluid volume, which results in decreased perfusion in tissues, and this also leads to the loss in salt water from the extracellular fluid. So this condition is usually treated by salt base or crystallite infusion. Okay, so they pump in a lot of saline into the person. So dehydration, on the other hand, is just the loss of water alone. Okay, and a person is said to be in hypovolemic shock when they lose more than 20% of their blood and the mean arterial blood pressure falls below 60. It's considered a severe hypovolemic shock. Okay, and when there's a serious damage like a hemorrhage and when a person goes into a hypovolemic shock, it cannot be controlled. And now when it cannot be controlled, the intrinsic autoregulation stops and the extrinsic mechanism take over. So when the blood pressure in general falls below 80, the extrinsic mechanisms take over and the intrinsic mechanism cannot be controlled or no longer compensate. So the extrinsic control mechanisms include the sympathetic nervous system and mainly the hormonal mechanism. Now, under extreme stress, the stress hormones are norepinephrine and epinephrine that are released by the adrenal medulla, and both of these are potent vasopressors, meaning they cause vasoconstriction of afferent arterioles, and they inhibit filtration because they constrict the arterioles. So they prevent a lot of blood entering the glomerulus. Okay, and by inhibiting filtration and constricting the afferent arterioles, they trigger the receptors around the juxtamedullary apparatus to release renin. Now, if you remember from the previous chapter, renin is released when the blood pressure is low, and they trigger the release of angiotensin, right? So renin, when it's released, it helps in lowering the glomerular filtration rate, but it affects the blood pressure differently. Okay, so keep this in mind. When the blood pressure increases, the afferent arterioles get constricted and the efferent arteriole gets dilated. Okay, and by constricting the afferent arterioles, you are reducing the amount of flow of blood to the glomerulus. Okay, and in the presence of norepinephrine and epinephrine, both of them cause constriction of afferent arterioles because both of these are also potent vasoconstrictors. Okay, now when epinephrine and norepinephrine get released, they constrict arterioles, inhibit filtration, and trigger the release of renin enzyme. So when all of this happens, the glomerular filtration rate gets lowered. They also cause the shunting of blood away from the kidneys, meaning they close the sphincters around the capillaries, and they prevent excessive flow of blood towards the kidney during extreme stress. Now this video over here, if you click on this link, it'll ask you, it'll show a link for animation, so you can watch the animation for extrinsic controls of glomerular filtration rate. Now, you have already studied the renin-angiotensin and aldosterone mechanism in the past. You've looked at it in the respiratory system, you looked at it in the cardiovascular system, and now again in the urinary system. Now, if you remember from the earlier slides, renin is actually secreted by the granular cells in the juxtamedullary apparatus. And these renin secretion from the granules respond to drop in blood pressure, any sympathetic stimulation, or low NaCl detection by the cells of the macula densa. So when the blood pressure in the body is low, the granular cells secrete renin, and the renin enters the blood plasma, and it reacts with the blood plasma proteins to form angiotensin 1 from angiotensinogen. Now, in the lungs and the blood plasma, ACE enzyme converts angiotensin 1 to angiotensin 2. Now, angiotensin 2 is the final worker that affects blood pressure. The first thing it does is to activate the hypothalamus to make us thirsty. So we drink a lot of water, which causes the blood pressure to increase. Okay, but the main job of angiotensin 2 is to cause vasoconstriction. Okay, so vasoconstriction involves constricting the blood vessels, meaning the blood vessels get squeezed like the water hose that we talked about. So when you squeeze the tip of the water hose, the blood pressure or the water pressure gets increased, right? So you can spray water a lot farther.
Okay, so vasoconstriction leads to increase in blood pressure. And then it affects the adrenal cortex, and the adrenal cortex stimulates the release of aldosterone. And aldosterone is a sodium and water retaining hormone. So aldosterone helps the body, or tells the body, to retain sodium, and water follows sodium. So when water and sodium gets retained, it leads to elevated blood pressure. So keep in mind, renin is secreted by the kidney, and make sure you know how renin affects angiotensinogen and how angiotensin II affects different parts of the body to elevate the blood pressure.